In this video, we'll explore the fascinating world of Genome-wide association studies, or GWAS, and their role in developing climate-resilient crops. We'll delve into how these advanced genomic tools are revolutionizing agriculture and helping us tackle the pressing challenges posed by climate change. The increasing unpredictability of climate patterns poses a formidable challenge to global food security. Rising temperatures, erratic rainfall, prolonged droughts, and shifting disease dynamics threaten the sustainability of agricultural systems, necessitating an urgent search for solutions that ensure crop productivity in the face of these adversities. Conventional breeding approaches, while historically significant, are often constrained by long selection cycles and a limited understanding of complex genetic traits. Modern genomics has revolutionized this landscape by offering powerful tools to dissect the genetic basis of climate resilience in crops. Among these, genome-wide association studies, or GWAS, have emerged as a cornerstone in the identification of genetic variations linked to desirable agronomic traits, providing the foundation for developing climate-resilient crop varieties. Genome-wide association studies, or GWAS, leverage high-throughput genotyping and statistical association analyses to detect genetic loci linked to specific traits. By comparing the genomes of diverse plant populations, GWAS identifies single nucleotide polymorphisms, or SNPs, associated with phenotypic variations in stress tolerance, yield stability, and adaptive traits. Unlike traditional quantitative trait locus, or QTL, mapping, which requires biparental crosses, genome-wide association studies, or GWAS, capitalize on natural genetic diversity. This provides a high-resolution approach to pinpointing candidate genes underlying climate resilience. A defining feature of GWAS is its reliance on large-scale datasets integrating genetic, phenotypic, and environmental parameters. For instance, the development of global crop panels such as the Diversity Panel of Rice, C4 Rice, and the Genome Analysis Toolkit, GATK, for maize and wheat, have facilitated comprehensive GWAS analyses. These efforts unravel the genetic basis of stress-adaptive traits. The success of GWAS hinges on the statistical power afforded by high marker density population structure correction and robust phenotyping. This allows researchers to overcome confounding factors that often obscure genetic signals. GWAS, or Genome-wide Association Studies, has emerged as a powerful tool in understanding how plants cope with drought and heat stress. By scanning the genomes of diverse plant populations, researchers can identify genetic variations that contribute to resilience against these harsh conditions. This knowledge is crucial for developing crop varieties that can thrive in the face of climate change. Drought and heat stress are among the most pressing climate-related constraints on agricultural productivity. In major staple crops such as wheat, rice, and maize, yield losses due to water deficits can exceed 50% under extreme drought conditions. GWAS has been instrumental in identifying genetic determinants that confer drought and heat resilience by pinpointing genes involved in osmotic adjustment, root architecture, and stomatal regulation. A landmark GWAS study on rice, or Oryza sativa, identified key drought-responsive genes, including deeper rooting one, which enhances root depth and water uptake efficiency. Similarly, a study in maize, known as ZIA maize, uncovered variations in the ZMVPP1 gene. This gene encodes a vacuolar H+ pyrophosphatase enzyme that enhances drought tolerance by improving cellular osmotic balance. In wheat, or Triticum aestivum, GWAS revealed that allelic variation in TADREB3, a dehydration-responsive element-binding protein, plays a crucial role in modulating drought resistance by regulating stress-inducible gene networks. Beyond these individual gene discoveries, multi-environment GWAS analyses have highlighted the polygenic nature of drought and heat resilience. The GOBI, or Genomic and Open Source Breeding Informatics Initiative, for wheat and rice has integrated GWAS findings with predictive breeding models, enabling the selection of high-performing genotypes adapted to water-limited environments. These insights are now being applied in breeding programs, accelerating the development of drought-tolerant cultivars with stable yields under fluctuating climatic conditions. Salinity and waterlogging are increasingly prevalent in coastal and inland agricultural regions, exacerbated by rising sea levels and erratic monsoons. GWAS has provided critical insights into the genetic basis of salt tolerance, particularly in crops such as rice and barley, which are highly sensitive to saline stress. One of the most well-characterized salinity tolerance genes identified through GWAS is OSH-KT1, 5 in rice, which regulates sodium ion transport and prevents toxic accumulation in leaf tissues. Similarly, a comprehensive GWAS on barley, also known as Hordeum vulgare, linked salinity tolerance to polymorphisms in HVNAX3, a gene encoding a sodium exclusion transporter. In soybean, also known as glycine max, GWAS identified GM-SALT3, a gene that enhances salt tolerance through improved ion homeostasis and oxidative stress mitigation. Waterlogging, another major abiotic stress, severely impacts crop productivity by limiting oxygen availability to root systems. A GWAS analysis in maize identified ZMADH1, an alcohol dehydrogenase gene involved in anaerobic metabolism, as a key determinant of waterlogging tolerance. These discoveries are now being integrated into genomic selection frameworks, allowing breeders to develop salt and flood-tolerant varieties suited for vulnerable agroecosystems. Climate change is not only altering abiotic stress patterns, but also reshaping disease dynamics in agricultural landscapes. Warmer temperatures and fluctuating humidity levels are fostering the emergence of virulent pathogens that threaten global food security. GWAS has been instrumental in deciphering the genetic basis of disease resistance, enabling the development of pathogen-resistant cultivars through marker-assisted selection. In wheat, GWAS has identified Lungyer67, a gene conferring multi-pathogen resistance against leaf rust, stripe rust, and powdery mildew. In rice, studies have pinpointed XA5 and XA13, two genes that provide durable resistance against bacterial blight, a devastating disease in South and Southeast Asia. A large-scale GWAS in maize uncovered novel resistance loci for southern cornleaf blight, a disease exacerbated by high humidity conditions associated with climate change. One of the most transformative applications of GWAS in disease resistance is its integration with genomic prediction models. By leveraging high-density SNP markers and machine learning algorithms, breeders can predict disease resistance profiles in early-generation breeding lines, dramatically reducing the time required to develop resistant cultivars. This approach is now being widely implemented in wheat and rice improvement programs under the SIMIT, the International Maize and Wheat Improvement Center, and the International Rice Research Institute. GWAS for nutritional security and carbon sequestration. Climate resilience in crops extends beyond stress tolerance to encompass nutritional quality and carbon sequestration potential. GWAS has played a pivotal role in uncovering genetic variants associated with micronutrient accumulation, particularly in biofortified staple crops. A major GWAS study in maize identified allelic variations in ZMVT1, a gene regulating iron and zinc transport, paving the way for enhanced biofortification strategies. GWAS in rice. Similarly, in rice, genome-wide association studies uncovered key loci associated with high beta-carotene content, a precursor of vitamin A, which is crucial in combating malnutrition in developing regions. Beyond human nutrition, Genome-wide association studies are also being applied to enhance crop contributions to carbon sequestration. A recent study in sorghum identified genetic determinants linked to increased root biomass and deeper rooting depth, traits that improve soil carbon storage and contribute to climate change mitigation. These findings are particularly significant in the context of sustainable agriculture and regenerative farming practices. Despite its transformative impact, Genome-wide association studies, or GWAS, faces several challenges in its application to climate-resilient crops. The complexity of gene-environment interactions often complicates the identification of causal variants, necessitating integrative approaches that combine GWAS with transcriptomics, epigenetics, and phenomics. Additionally, the accessibility of high-quality phenotypic data remains a bottleneck, particularly in low-resource settings where climate resilience is most urgently needed. Future advancements in multi-trait GWAS, which simultaneously analyze multiple stress tolerance traits, hold promise in addressing these challenges. The integration of AI-driven genomic selection and pan-genome GWAS, which incorporate diverse crop-wild relatives, is poised to accelerate the identification of novel resilience traits. Furthermore, the application of gene editing technologies such as CRISPR-Cas9, informed by GWAS discoveries, opens new frontiers in precision breeding for climate-resilient agriculture. Genome-wide association studies have redefined the landscape of crop improvement by unlocking the genetic basis of climate resilience. From drought and heat tolerance to disease resistance and nutritional security, GWAS has provided unparalleled insights into the genetic architecture of complex traits, empowering breeders with the tools to develop next-generation crops. As climate change continues to challenge global agriculture, the integration of GWAS with cutting-edge genomic and breeding technologies offers a pathway towards a more resilient and sustainable food system. Through continued investment in genomic research and international collaboration, the promise of climate-resilient crops can be fully realized, ensuring food security for future generations in an increasingly uncertain world.